are uniquely capable of complex cognition. Things like decision-making, problem-solving, and creativity are largely driven by a portion of the brain called the cortex, which is made up of these areas specialized for different functions. Development of these areas relies on carefully guided molecular cues that provide early instructions for how cells, layers, and areas of the cortex should be made. Here in the Bhaduri lab at UCLA, we use stem cell models to study the mechanisms of human brain development. My colleagues and I use organoids, 3D cell culture models derived from human stem cells, to study the processes governing brain development. Organoids generated from human stem cells replace the need for animal models in studying neurodevelopment. Organoids recapitulate programs of early human brain development absent in commonly used animal models, providing a refined system for studying uniquely human aspects of cell fate specification. I was initially interested in studying neuropsychiatric disorders, but I chose the subfield of neurodevelopment research because I realized there were so many unknowns about the basic mechanisms of neurodevelopment. In order to truly understand what goes awry in disease states, we need to understand how the brain is formed. The goal of my project is to understand how extrinsic inputs from the thalamus, a neighboring brain region to the cortex, influence cortical development. While in the adult brain, the thalamus is best known for relaying sensory information from the peripheral nervous system to the rest of the brain, it actually plays an important role, but often overlooked role, in development. Prenatally, the thalamus and cortex grow in parallel and send reciprocal projections toward one another. They use these long-range connections to communicate with one another to influence the growth and maturation of one another's neural stem cell populations. My mentor, Dr. Aparna Bhaduri, is on the cutting edge of the neurodevelopment field as she seeks to integrate 3D organoids with advanced next-generation sequencing methods. Under her guidance, I've been able to develop the idea to fuse two organoid types together into a comprehensive strategy to determine the extrinsic mechanisms governing cortical development. Because in a single batch, I can generate hundreds of organoid replicates, they can also be easily manipulated and screened for genes necessary to drive any observed changes in cortical cell maturation. As a recipient of the IFER Graduate Fellowship, I plan to highlight the utility of the organoid system to understand connections in the developing human brain. Previous studies in this field have relied on genetic editing in mouse and rat animal models. However, current in vivo models are confounded by the fact that they are unable to fully eliminate thalamic input to the cortex. These animal models also don't fully capture the diversity of cell types present in the developing human cortex. Organoids, on the other hand, can address this issue. The fusion of two organoid types provides a high throughput platform for visualizing cortical development in isolation or with extrinsic inputs. My entire life, I've always been an animal lover, and I try to embody this value in both my personal and professional life. I intentionally chose to work in the field of organoid research because I'm a strong advocate for animal welfare. I've been a vegetarian since high school and am a foster for an animal shelter in the Los Angeles area. I consider it a core part of my ethos to care about animals and our environment. I align with the goals and values of the International Foundation for Ethical Research, and through this fellowship, I will continue to incorporate this personal philosophy into my career aspirations and scientific practice.